let's get into it. Week 7, time passes fast. So this is webinar series number 7 already. And I'll be giving this one as well as the one next week. And so this time it'll all be about proteins. So Colin covered a lot of the DNA logic and the genetic systems. I'll be going over the protein systems. And so we're shifting gears now, going into a different language of amino acids rather than A, T, C, G. Now, today's talk is on enzymology. And some of you might know what enzymes are, or some of you might even have some experience in the field of enzymology. But today I just want to go over some of the basics of this field. As you can see, looking forward, we have the protein engineering webinar followed by assay design, although Colin, you said you might change that to bioinformatics, right?
Yeah, I'm going to.
Okay, awesome. So we'll post an updated one next time then. And in 3 short weeks, you guys will be giving your presentation. So keep that in the back of your mind, So let's see, should we have any hard deadlines for, well, I think we're good, right, Colin? I think, we can let them work through it themselves. And you have, of course, if any questions comes up, just let us know.
Yeah, if you're going to submit a video, do it like the day before. That's a hard deadline. Don't do it day of.
Yeah, I think that's good. Yeah. But keep this date in the back of your mind. So not October, August, August 27. That's the important date to keep in mind. So August 27, you guys are up. All right. So on to enzymology. Before we dive into enzymology, I've been talking about how we're doing protein topics in the next 2 weeks. And so I do want to first give a quick review of what proteins are. We went over this probably 3 weeks ago, but I do want to, re-instill in you guys what proteins are. Proteins are chains of amino acids. So you take your amino acids and you link them up through chemical covalent bonds. And this spaghetti string then collapses on itself. It folds into a certain way into a 3-dimensional structure that we call a protein. And that function of the protein is determined by the structure of the protein. That's a really common phrase you'll hear, structure determines function. Now, what determines the structure of the protein? It's the sequence of amino acids. It's kind of like a computer, right? How does a computer know to add or subtract or divide or multiply or do all these operations? It's the sequence of 1s and 0s in binary. So kind of a similar idea. The sequence of amino acids determines the structure of the protein, which determines the function of the protein. It's a very central idea. Now, proteins have, of course, all kinds of functions. You all know a lot of them already, right? We've gone over some of them. We've gone over, for example, hemoglobin. I think I mentioned hemoglobin in one of the earlier webinars. It carries oxygen around the body. It's pretty important. You've got things like ferritin, which can store iron ions, and you've got rubisco. Does anyone know what rubisco is? What is this weird thing, rubisco? Rohan says it's used in the Calvin cycle. That's exactly correct. Yeah. What does it do? It fixes carbon. I almost said it fixes Calvin. It fixes carbon. It's used in photosynthesis, something to do with CO2. Yeah, exactly. So rubisco is the enzyme that takes CO2 from the air and it traps it into other organic molecules, which is super important, right? I mean, plants do photosynthesis so that we can eat them, right? I mean, that's like the basis of all of our agriculture. Right, so rubisco is the enzyme that takes carbon from the air and sequesters it into the plant cells for sugars, for proteins, for all kinds of things. So rubisco is a really important enzyme, and we'll see what an enzyme is in a bit. But you can see they all have these very distinct shapes, right? Antibodies have a little Y shape. Rubisco has this kind of helical, ball-y shape. Hemoglobin has these 4 chains. They all have a really distinct shape that's used for their function. Now, does anyone know what this person's doing? It's a bit of an aside, but segues are weird. Does anyone know what this person is doing? Or what the profession is of this person? Seems old, right? Almost like a carving. What is he doing? Scribe, yeah, Scribe. He's a scribe. What scribes did back in the old days, I wasn't there. I say like I'm there, but no, back in the days, these scribes would take passages of text and they would copy it down letter by letter, and that was their whole job. That was their whole career. And ancient Egypt, scribes were super, super, high professions. They were like extremely well regarded scribes. Now, you don't have many scribes today, right? If you go around saying you're a scribe, people will look at you weird. Why? Because we have Control-C, Control-V. And so we can just copy and paste things very easily nowadays. And so, I mean, this took over, right? Now it's incredibly easy for us to copy and paste. No one scribes anymore. Why? Because it's so fast. You can just copy paste and it's done. You've made a copy of your original text. And so that's the whole idea behind what I want to talk about today. So knowing that, I want to ask you all a question. What's an enzyme? And as always, there's the URL there, polev.com/alentong472. What's an enzyme? Catalyzes a reaction to speed it up. Nicely said. It acts as a catalyst. So we see the same word again, catalyst, right? You might hear, in everyday language, oh, this was a catalyst for an event, or, historical catalyst. It's something that causes something to happen. It lowers the activation energy. So we see a lot of these vocabulary terms coming up that we'll learn about soon. These are all correct. Other ideas. It causes a specific biochemical reaction. I like this answer as well because it has bio in it, because there are inorganic catalysts. There's catalysts used in chemistry, synthetic chemistry. What's specific about enzymes is it's a biocatalyst. It's a catalyst in living systems. It lowers the activation energy and it converts substrate into a product. Yeah, so this is another vocab word, substrate. Substrate means reactant. It's a thing you're using to make something else. It's a protein. Yeah, most enzymes are proteins. Now, what's interesting is that some RNAs are also enzymes. We call them ribozymes. So it's kind of cool how there's different types of folds you can make to catalyze reactions. They're in living things. It can speed digestion and metabolism. These are all wonderful answers. But you see, a common thread is it speeds things up. Right? It's like the Control-C, Control-V of biology. It's made of amino acids, yeah, because most of them are proteins. So proteins are made out of amino acids. I'll wait maybe a couple of seconds for maybe a few more responses, but these are all great responses. And I think it gets to the crux of what enzymes are. Enzymes are, if it will move forward, enzymes are biocatalysts. They make reactions in living systems go faster. And I think that was one of the very first responses up there. Enzymes make reactions go faster, just like Control-C, Control-V. Now, here's a concept that some of you might have been introduced to in your chemistry classes. So the concept of equilibrium is when the concentration of your chemicals stops changing over time. So look at this reaction, right? We've got NO2 and we have N2O4. At time t equals 0, we have a lot of N2O4, this blue line here, a lot of N2O4, but over time, this goes down. There's less and less N2O4 because it's being converted into NO2. See, but over time, it reaches this thing called equilibrium, which is when the concentrations stop changing. It's a very central concept in biology and chemistry. It's the idea of equilibrium. What does it mean for a cell to be in equilibrium? I want to see if anyone can guess. If a cell is in equilibrium, what are its properties? What can we conclude? In other words, if a cell's molecules stops changing in concentration, we have a name for that. Can anyone guess? G0, that's a good guess. So G0 is a phase of the cell cycle, or phase of the cell cycle, in which the cell just stops dividing, right? Senescence. I wouldn't say that's true equilibrium though. Someone says homeostasis. I guess you have a set point, but again, it's not this idea of chemical equilibrium, true chemical equilibrium. Isotonic is kind of getting at the idea. That's more about, osmosis. But I'm thinking about if all the chemicals in this sack of cells is at equilibrium, yeah, the cell's dead. That's the term we use for a cell. A cell in equilibrium is a dead cell. That's the main takeaway, right? It's the reason why we eat. It's the reason why we excrete. We've got to take in new things and excrete out other things, because if we just leave things as they are, it will reach equilibrium, and we can't survive in equilibrium. A cell in equilibrium is a dead cell. So Rohan, you got it right. Right. So I mean, this is the reason why we need sunlight, for example. Right. Sunlight inputs energy to keep the plant cells from reaching equilibrium, to drive photosynthesis, to make sugars, which we then eat, to drive our own metabolisms. All of it has inputs and outputs. It's never a constant supply of a molecule. Now, of course, people have said about, people talked about homeostasis. That's true. But, homeostasis in the biological sense is that you have a set point, but that requires an input of energy. If you let it run for just, a long period of time, it will drop down to some number or rise to some number. It takes energy to maintain homeostasis. Right? Equilibrium means there's no energy, really, input or output. Equilibrium is just stasis. Okay. Now, what about when things are changing? That's when things are exciting, right? When a reaction happens, things change. Chemical bonds make and break. That's the idea of a chemical reaction. Now, when we talk about enzymes, we talk about reaction rates. We want to make things go faster. How do we determine the rate of a chemical reaction? So for example, in this graph right here, how do we determine the rate of the chemical reaction of turning N2O4 into NO2 from this graph? Rohan says derivative, and that's exactly right. So for those who haven't taken calculus yet, derivative means slope. That's literally it. You look at the slope of the initial point. So right here, the slope is about that. Right here, the slope is about that. You can say that this slope is the rate at which N2O4 is decreasing. And we can convert that rate into a reaction rate. It's really just the negative of that number. Okay? That's the idea. The rate of change of a chemical reaction is just the slope of the concentration as time passes at the beginning. Okay. Now, how do enzymes actually work? This is how enzymes work. I don't know why the enzyme's evil in this picture. I just found it online. Maybe it was Halloween themed or something. But you've got this, Dracula enzyme coming along. And then the Dracula enzyme finds a substrate. Substrate is another word for reactant. It's your input. So your input, your substrate reactant, goes into the enzyme and it fits into a pocket called the active site. Notice this is where the structure is really important, right? You want to have the active site shape fit the substrate shape. That then forms the enzyme substrate complex. The reaction happens. In this case, for example, the enzyme literally splits this poor guy in half. And then product release happens. And so this is how enzymes work. Enzymes bind, they catalyze, they make a reaction happen, then they release. That's the idea. Right? So if an enzyme makes a reaction go faster, we expect that an enzyme would make this rate in the beginning more steep. Right? The rate is increased if the slope has increased. Okay. Now here's a bunch of enzymes. And here are the crystal structures of the enzymes. In other words, people have taken these proteins and they've spent a lot of work crystallizing these proteins, and they shot X-rays at them to get these structures. They're called X-ray crystal structures. And they're all enzymes. Does anyone know what these are? ECO R1, ECO R5, PBU2. What are these things? Exactly. They're restriction enzymes, right? What are restriction enzymes? Notice the name enzyme is in that word, right? Restriction enzyme. They're enzymes that do what? It's a protein that cuts DNA, precisely. Yeah, cuts plasmids, cuts DNA, exactly. Do you see the DNA? Right there, that's DNA. So they've complexed this restriction enzyme with the DNA. They co-crystallized it, and they found out how the DNA actually fits into the active site of the enzyme. And you can see how beautiful this is, right? You see how you've got the enzyme with two subunits hugging the DNA in this kind of clasp. And they all have different structures. Why do you think the restriction enzymes have different structures? They look different, right? Why do they have different structures? I mean, they all cut DNA. Why the difference? So they can fit different substrates, bind to different sequences. Exactly, right. So even though the substrate is DNA, as Colin has talked about extensively, it's got different sequences. It can be ATCG, it can be GATTCC, right? All those sequences have distinct binding sites. So you can see how specific this is, right? It's almost like reading Braille. These enzymes, they scan the DNA double helix, feeling for the right binding sites. And once it feels for the right site, then it attaches, catalyzes, breaks the DNA, and then it can get to work. This is a really integral part of synthetic biology. You use restriction enzymes all the time. Now, here is an example of what they might do. So EcoR1 is an enzyme that cuts along the G and the A. So it cuts right between that, right? Same here, G and the A. Now, you notice here is GAATTC. Here is GAATTC. So it cuts right there and it cuts right there. Why do you think the sequence is the same forward and backwards? It's really common, actually. You see these palindromes all the time. It's the same forward and backward. Why is that? And I'll remind you back of the structure that people have so painstakingly solved. Right? Take a look at this. What do you observe? Why are they palindromes? Rohan says they can bind to either strand. That's true. There's another more fundamental reason, but you're right. They could bind to either strand. But here's the thing, they cut both at the same time, actually. It forms what's called sticky ends. So it cuts here and it cuts here. There's a really elegant solution, actually, you can do to figure this out. To bind to complementary strands, so these are complementary strands, that's true. But what's distinct about it being palindromic, right? GAATTC, backwards is also GAATTC. Why is that? And you want to think structure. Okay, so look at these structures and see what you observe. What do you notice? I'll give you a hint. What do you notice? Observations, anyone? They're symmetrical, right? They're symmetrical. It's a really elegant solution, right? Why do restriction enzymes bind to palindromic DNA sites? Imagine, you got one hand that feels for, it's literally like Braille. They feel for one specific sequence, and your other hand feels for the other sequence, but it's oriented in the other way, right? So it can feel for those palindromic sites by having these two clasps feeling along the DNA, right? So this strand might sense GAATTC this way, this strand might sense it that way. So it's homodimeric, right? So the structure is really important. And that's why. So it can sense these palindromic sequences and it cuts right where it needs to. Okay.
That's poetic. That's crazy. My mind's blown.
I think it's super cool, right? I mean, structural biology really is a really, rich treasure trove. And here's kind of zooming into those binding sites, right? You can see it's really complicated. But they've simplified it here to show you the interactions. For example, they've got hydrogen bonds using the arrows. They have the dashed line as the van der Waals forces. The whole idea is that these things bind with specific sequences, and they're really, really specific. honestly. They rarely cut in other places. They might have some leakiness, but you can see how it has all these interactions just to feel for the right site. Some even use magnesium, right? They have cofactors. They have other things required to bind. And it catalyzes this reaction. It cuts right there. They aren't showing the other one because it's identical. It's the other way around. So you can see how all these interactions, they require a three-dimensional structure to bind to DNA. Right? This is how enzymes work. Enzymes, they feel for the substrate, and once they feel it, they then, boom, they catalyze the reaction really fast. Okay, any questions about that? Okay, now someone actually in the beginning, they mentioned digestion. That's really important, right? I mean, when you eat food, when you eat a bunch of proteins, for example, fish and beans, they have a lot of protein inside of them. You eat it, goes down your throat, goes into your stomach, it's got to be digested, right? Your cells can't use the cells can't use the proteins from a fish directly. You got to take the proteins from the fish, digest the protein into its constitutive parts, and then make up your own proteins from those parts. So when you eat, this is what's happening inside your stomach. When you eat proteins, your proteins, they unfold, first of all, then they get cut. They get cut and cut and cut and cut. That's a chemical reaction, right? So when a protein gets cut, you're breaking chemical bonds. And so you can imagine if it takes a long time to do that, you might not be able to digest really fast. So I want to walk you guys through a thought experiment, right? So let's say you eat some fish, or you eat some salmon, or you eat some beans, and you eat some protein. It goes into your stomach, and let's say you don't have an enzyme. Okay. Let's say your stomach is a neutral solution for thought experiment purposes. It's neutral. It's at room temperature, and people have measured the rates of that reaction. Let's say you have a typical bond in the protein. This is a diglycine. Glycine, that's two amino acids formed together. And here's some chemistry going on. I don't want to get too deep into it, but the idea is you take this, one amino acid, two amino acids, and you break it up into these two. Right? That's the whole idea. It's digesting this dipeptide. This diglycine turns into two monoglycines. That's the chemical reaction. So this is what's, an example of a protein peptide bond. Here's my question to you. If you were to eat this protein, how long do you think it'll take? What's the half-life of this reaction? In other words, how long do you think it will take for half of this protein to be digested? If you just throw it into water, room temperature, what's the rate of that reaction? What's the half-life of the reaction? 3 hours. Okay, someone guessed 3 hours. If you throw it into water, it'll take 3 hours for half of it to digest. Break in half. Other guesses? Neutral solution, neutral, right? So no acid, no enzymes, no anything. 2 hours. Other guesses? Half-life means what's the time it takes for half of the reaction to be completed? 4 hours. I see we're in the hour order of magnitude. Other guesses? Half an hour. Okay. 30 minutes. 2 minutes. Okay, we're getting really fast now. 2 minutes. So for example, if you had 100 of those molecules, how long would it take for 50 of them to break apart? That's the question. Rohan asked, is this with enzymes? No enzymes. So this is a neutral solution, room temperature, no enzymes. Without a catalyst, how long do you think it'll take? That's my question. 10 hours, okay. Still in the hour order of magnitude. Other guesses? Three hours, okay. I want someone to be really brave. 5 years, okay, now we're really big now, 5 years. 1 hour, we got one outlier there, I see. 10 minutes, okay, we're in the minute range, the year range. 2 days, okay, so we have a day range in there. 2 days. Colin, you have a guess?
A lifetime.
A lifetime. Let's say 100 years, right? 100 years. 6 months, okay, half a year. You want to know the answer? Come on, let's go. Never. 350 years. If you had 100 molecules of this protein, of this dipeptide, it would take 350 years for 50 of them to be digested with no enzyme. Imagine if your steak took 350 years to digest, only half of it, right? That's just ridiculous. So you can see why we need enzymes, right? Reactions don't really happen fast enough for life to happen unless you have catalysts, unless you have enzymes, right? So I mean, you eat food and it might digest in an hour, a couple minutes, right? It depends. But with no enzyme, it just absolutely, you just can't imagine life without it. So this just really highlights the importance of enzymes. They speed things up, right? From 350 years to on the order of minutes or hours. That's many orders of magnitude difference. Okay, and here's just a list of all, not all, but some of the numbers related to these enzymes. Now these numbers really blow my mind. So look at some of these numbers. I don't want to really get too deep into the what they mean here. We'll get into that in a bit later, but just look at the rates, all right? For example, cyclophilin, it goes from this number to this number once you add the enzyme, okay? 4.6 times 10 to the 5, that's like what, almost a million? Almost a million times faster, okay? This one, almost, over a billion times, sorry, yeah, over a million times faster. This one's, over a billion times faster. This one completely just blows me away. This one is almost a billion times faster. These are numbers that I can't even wrap my head around, right? So enzymes are super important. They can make a reaction that doesn't really do anything go fast, 39 times per second, right? Like that, crazy fast.
There's a question in the chat.
Let me see. Can you speed up the rate by changing the enzyme? Yes. Yes. And so if you have a different enzyme, it might catalyze it faster. Some enzymes are better, some are slower. And that's kind of the reasoning behind enzyme evolution too. I mean, people study how enzymes evolved. They went from slower to faster and faster and faster. Sometimes you might even want it slower. Maybe there's a regulation process that you might want it to be slower. And people are thinking about synthetic biology and how do we change the enzyme, mutate the enzyme through the DNA modification? How do we make it faster? So those are all questions you can ask. But yes, the enzyme can change, and that can change the rate for sure. Yeah, Colin?
I think Rohan asked it, chat, but explain what K-cat, K-non are.
Oh, yes. So I have a slide on K-cat coming up in the future. K-non is the rate constant for a chemical reaction. Let's see. I don't really go into the non-enzymatic rates, but go to any AP Chem class and they'll talk about the rate constant of a reaction. And so the idea is if you have, for example, a first order rate constant, it means the more reactant you have, the faster it will go. That's the idea. And so, you can draw a graph like, here's the rate and here is the amount of substrate. As the amount of substrate goes up, R goes up, right? More substrate, the faster it goes. The slope of this is K, that's it. The slope is just K non. Now, Parsa asked, can these be modeled mathematically? Yes, the answer is yes. I'll get into that in just a bit. But other questions before I move on to like the more math heavy version. Okay, but I think the main takeaway is, don't memorize these numbers, but just appreciate how fast it enhances these rates. I mean, these are mind-blowing numbers. I mean, you guys might not, think, 10 to the 17, that's like, what, 1 with 17 zeros after it? Ridiculous. All right, let's move on. There's a bunch of types of enzymes. I won't get into this, but there's different types of enzymes. You can categorize them. But I do want to get into some of the math. But before I do that, I want to show you guys a map of metabolism. So while I'm going through the different parts of this map, you can see how crazy it is, right? While I'm going through the different parts of this map, maybe just like type in the chat what you notice, what you see, what you know, what you have heard about or have, just type out observations in the chat as we go along, okay? So I'm going to just maybe, it's a Google Maps style sort of map. People spend a lot of time trying to figure out these pathways, but just type in what you see, what you know, right? So we have the glucose. Let me pull up the chat actually while I do this. Many lines, right? Seems like a headache. This is biochemistry, right? So there's a lot of complex systems going on in this. So, I mean, many of you probably have heard about cellulose in paper, right? Glucose, the sugar. You see a lot of these chemicals, a lot of structures. What do you notice? Lignin in wood. That's how you make wood, right? Does anyone recognize what this is? What these are? Looks like a honeycomb. Yeah. A lot of structures, a lot of hexagons. Right, ATP is energy, right? And you can see how it's used to make DNA and RNA. It's kind of cool. So this is how you make DNA in cells. It's the metabolism to make DNA. Folic acid, right? Vitamins, glycine, what we were talking about before. You have sugar metabolism, you have lipid metabolism, fatty acids, you have mitochondrial metabolism, more fatty acid metabolism, amino acid metabolism. You can see there's so much stuff, so much stuff. I mean, you can spend your whole life studying a single one of these reactions. Now, next to each of these arrows, you see a number. And each of those numbers is an enzyme. Look at all the enzymes inside your cell. Think about how complex this is, right? This whole system has to work together to make you work. You can see all these numbers. For example, here you have a succinyl transferase. You see here there is, what is this? This is a hydratase, right? That's in respiration, actually. You see here we have an acetyltransferase, another ACE, right? All these ACEs, when you see ACE, phospholipase, all these ACEs mean enzyme. So in the future, when you see the word ACE, just think enzyme. Oh, sucrase, that means it breaks down sucrose. It's an enzyme that breaks down sucrose. Here we have a diesterase, cleaves phosphodiester bonds. All these ACEs, they represent enzymes. Okay, so just not to scare you from biochemistry, but just to show you the complexity of the whole system, which I find kind of cool. Okay, just to zoom out again, just to see the whole thing. Okay, this is life. Life is chemistry. You might want to hide from it, but you can't. Okay, life is chemistry. All right. Now, before going on to the nitty-gritty mathematics of all this, I want to do another thought experiment. So let's say I'm here. I'm that dot. And I want to get, let's say here. Okay, somewhere on the other side of the ridge. Can someone draw for me a path that they would want to take to get from the dot below to the dot above? You should be able to annotate, I believe. I think I turned on annotations. So someone just draw on the screen, where do you think? Okay, so someone drew a path, you go down this road, it's nice. Someone goes above the hill and to the destination, great. Someone else drew a similar path, but they went on the little crevice, right? And then they went to the destination. All right, someone put one that's similar, but they went a little bit of the side route. And then they went to the destination. I'm gonna make a really wild one. I'm gonna go all over the mountain and then go to the destination, right? Some people might go this way, some people might go that way, right? Some people I call in are really, they're hiker energy. Now the question is, which one of these is the best one to take? Love it, love it. Some people just love hiking. I would argue that if I was, if it was me, I would choose this path here, the black one. I would go from the starting point, I would go down the nice path, very leisurely, go down the road and go to the place where there's the lowest hump. See that? And then I get to my destination. That's my preferred path. And I think you all can understand why, right? It's hard. Hiking's hard. Hiking's difficult. And so if you go like this, it's a lot less energy needed. Now, this is a similar logic that we take when we talk about chemical reaction. Okay, let's say you want to get from point A to point B. Here's the reactants, here's the products. And these axes might seem weird to you. They're just, geometry. This atom might move here, might be bond angles, might be bond lengths, whatever you choose. Right? We call them reaction coordinates. But you might want to go from A to B. You don't want to go to these high points. You go to the lowest hill, then you go down. Okay? And that's what nature does. So you assign an energy, just like in the previous example, you assign an energy to all these points. And the ones that are high up, they have high gravitational potential energy, right? Some have lower gravitational potential energy. Same thing for chemicals, right? If you have a certain arrangement of atoms that might have high energy or low energy, same idea. It's a landscape of energies. And you, as I guess the abstraction of a reaction, want to take the path that has the lowest hump. Very easy, because it's the easiest path. And if you imagine taking a cross section through this three-dimensional surface, then you might get something that looks like this, right? You might get some point A to point B, and you go over some hump to get to where you want to go. Oh, I see someone has a question. Oh, looks kind of like gradient descent. Yeah, so it's similar, right? Although I guess in this case, it's not really descent, it's like going up, but then you descend. So that's a good one. So reactant, product, you gotta pass some hill to get to it. This is something you'll see over and over and over again in chemistry. All right. Now that height, when you get from reactant to the very tippity top, that height is called the activation energy. And someone brought it up in the very beginning. Enzymes, all they do is they lower the activation energy. They do this. Why is this easier? Or I guess, why does this make it faster is my question. Thoughts? Why would lowering this hump increase the rate of the reaction? Enzymes do this. Enzymes just, all they do is just lower the hump. It's almost like they TNT the hill and they make a pathway through in which it's easier to cross, right? So why does that make it faster? Thoughts? Because the more energy required means that more work is needed. I think that's getting to the point, right? If you have a higher hump, you need to input more energy to get it over that hump, right? If you need to input less energy, say for example, just the thermal jiggling of the molecules, then that's enough to get it over that hump. Then you've got an enzyme on your hands, or your enzyme has catalyzed the reaction enough to make it faster. So that height of the hump is essential for the rate of the reaction. The lower the hump, the faster the reaction. Someone said it's more tiring to climb up a mountain than the flat path. Exactly. It's all about energy. Now I'm going to dive into a little bit of the math behind it. This is a really famous model you might hear about in your biochemistry courses. It's called the Michaelis-Menten model. And this is what they thought about. They thought, well, an enzyme has to first bind to a substrate, right? In order for an enzyme to work, it's got to first hit it. It has to contact it in the active site. So that's this very first step. Enzyme plus substrate makes the enzyme substrate complex. It's just them two bound together. And then the enzyme does its thing and it releases the product and it returns the enzyme. So notice the enzyme wasn't consumed, it's remade again. So this enzyme can literally go back and do its thing all over again. It's recycled, right? So this is the model, the very simple model that Michaelis and Menten came up with. Enzyme plus substrate, they go back and forth into a binding mode called the enzyme substrate complex, and they turn into products. Very simple. Now, Rohan, this is what you were asking about the Ks. The Ks represent the rate constants of these values. So if K1 is really big, that just means it goes there really fast. That's the whole idea. Now, I don't want to get into all the mathematics, but we do want the result. We want to understand the result of all their mathematical modeling. So they take this model, and this is where some calculus might be involved. very simple calculus, but there's still, derivatives. But they take this model and they bring it through a bunch of analyses and a bunch of assumptions. And the important part that we care about right now is the result, this equation right here. That's called the Michaelis-Menten equation. If you want to learn more about the derivation of it, I highly encourage you to go online, search it up, or watch this video afterward, try to piece it together. But what we want to understand is what does this model have to implicate for enzyme reactions? Just some terminology here. V, I guess it stands for velocity, but it's more like rate. Think of it as reaction rate. We talk about enzyme kinematics. Those of you who might have taken physics might have done kinematics, like rate and speed and acceleration, velocity. I guess biochemists just borrowed the V. So V stands for the reaction rate. V0 is the initial reaction rate. Remember the beginning, the slope of that beginning? That's V0. So V0 is that slope in the beginning. Vmax is the maximum rate of the reaction. S is the concentration of the substrate. Km is a number, just a number. And all these things come together to make this final understanding of a basic enzyme model. This is the simplest one there is. Okay. V0 equals Vmax times substrate all over Km plus substrate. I can just spill it out, but what does it mean? Right, let's understand what this means. So I want to use this website called Desmos. I love Desmos. I use it all the time. I think it's great. So I'm going to pull up a Desmos tab. And I'm just going to put in the equation, just the Michaelis-Menten equation. V is equal to Vmax times the substrate, all divided by Km plus the substrate. Okay. Now I'm going to put in my parameters, Vmax and Km, and I want to focus on the positive region right here. Okay. So just to orient you all, the x-axis is the amount of substrate. It's basically the amount of reactant you have. And the y-axis is the reaction rate. I'm going to set these to 0 because 0 really is the limit. So watch what happens as I increase Vmax. I want you to type in the chat what you observe happens as I increase Vmax. Okay, you ready? Type it in the chat. What's happening? As I increase Vmax, what's happening to the plot? Right, as I decrease Vmax, what's happening? As I increase Vmax, what's happening? Zarek says, as Vmax goes up, the slope stays constant. Now, what do you mean by slope? Do you mean this first part or this last part? Oh, the last part. Yeah. So what Zarek is saying is the last part seems like it's plateauing. It's flattening out. So remember, the substrate concentration on the x-axis, the y-axis is the rate of the reaction. Now, that's a really good point. Why do you think it plateaus? Anyone can answer this. Why does the enzyme reaction rate plateau as you increase the substrate concentration? Let's zoom out. Notice, as I zoom out, it really plateaus to Vmax. Vmax is 10, it plateaus to 10. It asymptotically approaches 10. Rohan says, all enzymes are occupied. Exactly. It's saturated, right? And you'll notice that this is very similar to what Colin showed you, that graph, right? That sigmoid or the hyperbolic graph. This is what it is, right? So it's a saturation curve. If all the active sites are occupied, then the enzyme is working max speed, right? Max rate. It really can't go any faster. That's why it plateaus. Okay. So you played with Vmax. Let me set this to 100, let's say. 100. Okay, let's go even higher. There we go. So Vmax essentially sets the maximum rate, and it plateaus. The slope goes to 0 as it plateaus to that Vmax. Now watch what happens as I increase Km. Okay, again, type in the chat what you observe. As Km increases, what happens? What do you observe? They go from here, up, 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 up. What does Km mean? Can someone tell me? Rohan said it takes longer to saturate. It's a longer time needed to reach Vmax. Exactly. So if Km increases, it takes longer to saturate, right? You need a lot more substrate in order to reach the maximum velocity, the Vmax, right? In other words, it's the affinity of an enzyme for its substrate, right? What does a low Km mean? A low Km means that it takes very little substrate to get a high speed. It has a high affinity for that substrate, right? A really high Km means you got to take a lot of substrate to get that velocity up. You know, it's a very low affinity enzyme. So you can characterize these enzymes with Km and Vmax, and that's the simplest model of enzyme kinetics. So Zarek asks, the more substrate means that it takes longer to reach Vmax. The x-axis is the substrate amount, right? So if I have a high Km, you can see how it takes more substrate to reach Vmax. That's your question. If I have a low Km, you know, a little bit of substrate already gets it pretty high up. So a plateau is a lot sooner. Does that answer your question? I'm not sure. Or maybe Zarek, if you rephrase your question. Okay, awesome. Let's see, I've got maybe 5 minutes left. So I do want to show you guys something else. I didn't go over all my slides, but that's okay. It always happens. I want to show you something else that I think is really cool. So far, we've been dealing with a lot of assumptions. Right? Michaelis mentioned they made this model, this really beautifully simple model of enzymes. And they said, okay, if these things happen, you get this curve. It saturates. Awesome. Right? Now, a really simple modification can make this seem actually, you know, a lot more exciting. And here's how it works. When you assume Michaelis-Menten kinetics, you're really assuming a lot of things. You're assuming it's got one substrate. It's got, you know, one subunit really, and it you know, equilibrates between the ES complex really fast. All these assumptions are you know, maybe valid, maybe not. Now, if you generalize it one step further, you get not the Michaelis-Menten equation, but what's called the Hill equation. I think Colin mentioned it last time. It's actually really simple. This is what you do. You take your substrate and you raise it to a power. You raise it to, I don't know. Oh, nope, not that one. You raise it to the power of, let's say, a number n. You take your Km, you raise it to a power n. Take your substrate, raise it to the power n. All we did was we just raised certain numbers to a power of n, which we call this random coefficient. We call it the Hill coefficient. And n is 1. Sure, awesome. Gee golly. Now, watch what happens as I increase this Hill coefficient, okay? It's a modification, or I guess a generalization of the Michaelis-Menten equation. Again, type in the chat what you observe. As I increase n, watch what happens. Ready? It's actually kind of cool. Ready? What happened? I'll go back. I guess I can play it like this. I can, I'll go from 1. I can play it. Watch this. Yeah, there we go. Look at that. Automatic mode. So what's happening as n changes? Can someone tell me? What do you observe? Someone type something. It becomes more logistic. Yes. What does that mean? Logistic. It plateaus sooner. Yes. Yes. That's true. But I guess you can change the plateau sooner with Km, right? So that's a good point. That's a really good observation. But you can change the plateauing, you know, sooner or later with the Km value. What significance does the n have here? I think Rohan's close, but I want you to use, you know, more simple language. What does becoming logistic mean? The derivative changes a lot quicker. Yes, yes. More S-shaped. Yeah, exactly. It's more S-shaped. It's sigmoid, it's logistic. What it means is as n increases, it's becoming more switch-like. Colin said it last time, right? On, off, right? If you have a really high Hill coefficient, notice how it's just on, off. A really small change in the substrate concentration causes a really big increase or decrease in the rate of the reaction. That's super duper important for these kinds of regulation processes, right? If you want something that's really switch-like, you want something that can change fast, that's really sensitive. Right? So these models are really important for thinking about how these enzymes work and how they respond to environmental changes. right? On the other hand, if you want something that doesn't really change, you want a low Hill coefficient, right? A more gradual change. Maybe it might be, for example, I don't know, if you, I'm trying to think of an example, you know, if you are trying to, I don't know, as you grow or something, you know, if you try to model, for example, your growth, right? You might have a growth spurt, and then it might, you know, plateau as you get older. It might even get shorter as you grow older. You might be able to model that with these kinds of equations. Of course, that's an example. You know, you can't really do that with enzymes. But what's a good example? I'm trying to think of glucose. You eat a meal, right? After you eat a meal, you want your body to start being able to prepare itself for being able to metabolize glucose. You want it to be fast. You don't want your glucose to hang around for too long. And so you want it to be kind of switch-like. So these regulation pathways are pretty important. Okay. Any questions about the Hill equation, Michaelis-Menten equation, and what this curve actually means? Okay. In that case, let's see. I think I will actually end it there a little bit early for today because I'll get into this next time. I think it's pretty important. But there's our friend Rubisco, and we'll compare all these values next time. I think it's kind of cool how you can see, you know, the range of all these enzyme parameters across a lot of enzymes in biology. But we'll get into later and how we engineer these enzymes in all kinds of ways. But I think I will end it right there. All right. Any questions about today's webinar, about enzymes, about Michaelis-Menten equations, about Hill coefficients, anything at all? All right. Okay. I mean, hopefully you all enjoyed. It is 7 p.m. So if you have to go, you're free to go. Feel free as always to stick around for questions. And yeah, thank you all for coming.
See y'all.
But oh, someone had a question. So someone asked, how do you determine Km?
Yeah.
In that case, how do you determine anything at all? Right? How do you determine Kcat? How do you determine Km, the K1, K-1, and you know the K2? How do you measure all that? Experiments.
There's another really good question in the chat too. Or how to use synthetic biology to affect enzymes Vmax, or I guess. Yeah.
Yeah, we'll do. So I guess I'll answer the first one first. How do you determine Km? You can do enzyme assays. And so if you know the reaction enzyme catalyzes, say for example, it turns A to B, and you can measure how much B appears over say a minute or half an hour or an hour, you can measure that rate. You can do an experiment in which you increase the amount of A to see how the rate of B increases, and you can make that curve yourself. You can fit a curve to that graph, and you can measure Km that way. That's the most common way that you can do it. And yeah, Colin and I will try to do that actually, hopefully, when we get our enzyme for DMJ Hamty. And Zarek asks, can we use synthetic biology to increase an enzyme Vmax value? Yes. Yes. In fact, stay tuned for next week. I'll talk about Rubisco. That enzyme in the very beginning we talked about. Rubisco is an enzyme that fixes carbon from the atmosphere. Super important for bioproductivity, agriculture. If we can increase Rubisco's Vmax, or lower its Km, for example, that can help solve food shortage problems all over the world, which is super amazing. So the answer is yes. The short answer is yes. If we can increase an enzyme Vmax, then we can increase productivity. We can make more product. Yes, definitely. That's a really good question.